I'd like to take a second to introduce our seminar speaker. Our speaker is Dr. Jamila Roth. She has a bachelor's degree from Skidmore College and a very newly minted Doctor of Philosophy degree from the University of Florida. She graduated last month. She is a biologist, and she has some she's a biologist right now at the University of Florida, but we will lose her in a couple of months when she starts a postdoctoral scholar position at Mote Marine Lab. Jamila has a lot of broad and really applicable interests to the folks interested in the Nature Coast Aquatic Preserve. She works with marine plants. There's lots of marine plants within the preserve. And she's interested in how increased temperatures will impact plant-animal interactions, and particularly interested in how those changes in plant-animal interactions will vary across a stressor landscape. So how they'll vary when there's high nutrients versus low nutrients. She's keenly aware that some seagrass systems are more vulnerable to stressors than others, and she's really interested in understanding the mechanisms behind ecosystem resilience, which is what we will hear about today. This work really pushes the boundaries of our theoretical knowledge, which is great, but it also is really applicable to conservation and to ecosystem restoration, which is important when we're thinking about this preserve. And it's also really evident in the fact that her new position will be really focused on ecosystem restoration. So thank you, Jamila, for speaking with us today. And I'm going to go ahead and turn this over to you.
Cool. Thank you so much for the introduction, and thank you everyone for joining. You should be able to see the screen or my PowerPoint now. So today I'll be talking about a project from my dissertation research where I investigated how macrophyte species richness impacts resilience. And so, as I'm sure we're all aware, global temperatures are increasing. And you can see in this graph on the x-axis is the year, and on the y-axis is the temperature change in degrees Celsius. And so this black data shows historical trends, and then when the lines split into different colors, these are different scenarios with different greenhouse gas emissions. And so under a scenario with moderate to high greenhouse gas emissions, we're expecting to see a 3 to 4 degrees Celsius increase by 2100. And these temperature increases directly impact seagrasses. So they alter seagrass photosynthesis and respiration rates, as well as seagrass nutrient uptake and biogeochemical conditions. And so this can lead to changes in seagrass growth rate, and heat stress can even lead to seagrass die-offs. And warming temperatures also indirectly impact seagrasses. And one way they indirectly impact seagrasses is through tropicalization, which is the range expansion of tropical species into subtropical regions. And this alters food web dynamics and trophic interactions. And so tropicalization is occurring in many regions globally. So in parts of Japan, Australia, and the Mediterranean, tropical herbivorous fish have expanded their range into subtropical regions, which has led to overgrazing of the foundation species, which in this case is macroalgae. And this has led to a loss of the macroalgae in many regions, which has resulted in a shift to a barren ecosystem. And with this loss of foundation species, we also are losing really valuable ecosystem services. And so in the northern Gulf of Mexico, we're also seeing an increase in herbivorous fish populations, but we're less sure of how this increase in herbivory might impact the seagrasses in this region. And so in the northern Gulf of Mexico, we have one of the largest contiguous seagrass meadows in the continental United States, and it provides many really valuable ecosystem services, including habitat for fishery species and endangered species, carbon sequestration. It can serve as a denitrification hotspot, which removes excess nitrogen from the system, and it can also dissipate wave energy, protecting coastal communities from storms. And these seagrass meadows also contain multiple seagrass species that grow together within the same meadow. And as a result of tropicalization and conservation, we're also seeing increases in the abundance of manatees, green turtles, and emerald parrotfish in this region. And these are all seagrass herbivores. So this is expected to result in increased grazing pressure on the seagrasses and a shift from a detritus-based ecosystem to an ecosystem based on the direct consumption of seagrass. And so researchers predict that this will alter the seagrass ecosystems, perhaps resulting in less seagrass and perhaps resulting in shorter seagrass that resembles a mowed lawn. And so I'm interested in understanding the factors that might impact seagrass response to disturbances such as this increased grazing pressure. And so a lot of research has gone into how seagrass genetic diversity impacts response to disturbances. And there are a couple of reasons why seagrasses have served as a model organism for understanding the role of genetic diversity and resilience. And so seagrasses reproduce both sexually and asexually. And this asexual or clonal reproduction can result in the production of seagrass shoots that have the same genetic material. So they're genetically identical. And so researchers can propagate these clones in order to experimentally manipulate genetic diversity. And a lot of these studies have also been conducted in temperate regions where there's only one seagrass species in the meadow. And so this really facilitates these studies of genetic diversity since it removes the impact of species diversity. And so these studies have found that seagrass genetic diversity is really important and it can increase ecosystem stability and ecosystem functions. And so ecosystem stability has two main components. There's the resistance, which is the ability of the ecosystem to remain unchanged following a disturbance. And then there's resilience, which is the ability of the ecosystem to recover following a disturbance. So this resilience would be more the speed of recovery. And these studies have found that genetic diversity can increase seagrass resistance, it can increase seagrass restoration success, and it can increase seagrass resilience, with this increased resilience attributed to differential mortality, facilitation, and niche complementarity. So these different seagrass genotypes have different traits and different specialties, which allow them to interact positively rather than negatively. So if we take a step back from seagrass and think about a school setting, we wouldn't want a middle school to only have 8th grade history teachers, for example, because then the school wouldn't be functioning as intended. But instead, if there's different teachers with different specialties, the school would be better functioning and more effective. So it's the same idea with seagrasses, where when the seagrass clones have different traits and different specialties, they respond differently to disturbances and the ecosystem is functioning better. And so researchers have found that seagrass genetic diversity increases ecosystem functions. And so in this graph, in these figures on the right, you can see results from a study by Reynolds and colleagues where they measured allelic richness, which is a measure of genetic diversity. So high allelic richness means high diversity. And then they measured a variety of response variables, including productivity, measured as growth rate per area per day, as well as invertebrate density in the seagrasses, and then seagrass shoot density. And they found that allelic richness resulted in higher productivity, higher invertebrate density, as well as higher seagrass shoot density. And so similar to seagrass genotypes, seagrass species also vary in their traits and their response to disturbances. So we would expect to see similar patterns with seagrass species diversity that we see with seagrass genetic diversity. However, a lot less is known about the role of seagrass species diversity in response to disturbances. And this is partially because many of these studies have focused on seagrasses in temperate regions that only contain one seagrass species, while seagrass meadows in more tropical regions also often contain multiple seagrass species. And so there was one study that looked at the impact of seagrass species richness on restoration trials in Indonesia, and they found that seagrass species richness shown in these graphs on the x-axis resulted in increased survivorship and increased change in percent cover per month. And so like I said earlier, seagrasses in the Gulf of Mexico, these seagrass meadows contain multiple seagrass species. And so there's Halophila engelmannii or stargrass, Haliduli wrightii or shoalgrass, Syringodium filiforme, which is manatee grass, and Thalassia testudinum, which is turtle grass. And these species vary in their traits and their life history strategies. And so further on the left of this diagram are early successional species that are pioneer species. And so they usually have faster growth rates, but shorter lifespans. And then as we move to the right on this diagram, the plants are more persistent, so they're longer lived and they have slower growth rates. And these traits also impact their response to disturbances. So we expect that these pioneer species would have lower resistance, but a faster ability to recover, so higher resilience, whereas these species like Thalassia that are longer lived are expected to have higher resistance, but a slower ability to recover, so lower resilience. And since these different seagrass species vary in their response to disturbances and vary in their traits, we expect there to be higher stability with higher species richness. And the insurance hypothesis can be used to explain why we expect to see higher stability with higher species richness. So in these different plots, you see a simulated data for ecosystem productivity. And so in this first panel, there's species richness of 1, and then there's species richness of 2, species richness of 10, and species richness of 100. And then on the x-axis, we have time, and on the y-axis, we have productivity. And so the researchers assumed or said that the productivity for each species could vary between 0 and 1. And so when there's only one species, the productivity fluctuates a lot. So there's a lot of variability with large swings in productivity, and the mean productivity is around 0.5. And this is likely because the environment is changing, and this changing environment can have negative effects on this species. And then when we add a second species to have a richness of 2, we see that this variability decreases and the average productivity increases. And so this is because these species are fluctuating asynchronously. And so when the environment negatively impacts one species, it might have no effect on a different species. And so these different species can compensate for each other, and the variability can balance out to ultimately reduce these swings in productivity and increase stability. And then the pattern becomes even stronger when you look at a richness of 10 or richness of 100, where the variability is getting even smaller and the average productivity is approaching the maximum of 1. And so when there's even higher species richness, there's a greater likelihood that there would be species that are unaffected by a negative environmental condition, or that are not negatively affected by the environmental condition, which would compensate for species that might be more sensitive. And seagrass species can also have positive interactions with each other. So taller seagrass canopies can reduce photoinhibition of more sensitive understory species. It's also been observed that Thalassia canopies can prevent syringodium leaves from breaking, and having differences in below ground morphology can yield multiple benefits as well. So these varying morphologies can better stabilize sediments, which reduces nutrient release and prevents seagrasses from being uprooted. And having different rooting depths can also reduce competition for nutrients. So the larger seagrass species that have deeper roots are drawing from deeper nutrient pools when compared to the smaller seagrass species with shallow roots, which are probably using nutrients located closer to the sediment surface. And these diverse seagrass assemblages can also increase habitat complexity, which would provide better protection from predators for invertebrates and lead to increased invertebrate abundance. And invertebrates are really important for the seagrasses because they consume algae on the seagrass leaves and in the water column. And so this can reduce the shading of seagrasses by algae and buffer the negative impacts of nutrient additions on seagrass, ultimately increasing seagrass fitness. And so I wanted to explore how seagrass species richness impacts ecosystem stability in an established seagrass meadow in Florida. And so I conducted a field experiment in Crystal River, Florida, specifically working in a seagrass meadow located off the coast of Sandy Hook Island here. And so here you can see the seagrass meadow where we were working. So it was really dense and expansive, and it supports a lot of life. And you can see a couple different seagrass species here. So these wider leaves are the Thalassia testudinum or turtle grass. And then these narrower leaves, which are actually circular, are Syringodium filiforme or manatee grass. And so I asked a couple different research questions. I wanted to better understand how the response to grazing varies based on seagrass species identity and how macrophyte species richness impacts resilience following disturbances. And I specifically used a grazing disturbance since grazing pressure is expected to increase due to tropicalization. And so I set up 15 centimeter by 15 centimeter plots in this seagrass meadow. And so I had plots that only contained one species. So I had plots that only had Thalassia testudinum, only had Syringodium, or only had Haliduli, as these are the 3 dominant seagrass species. And then I had plots that initially contained multiple species. So they had 2 species, 3 species, or 4 species initially. And for the multiple species plots, they could contain any of these dominant seagrass species, or they could contain Halophila engelmannii, the smaller understory seagrass species, or Caulerpa prolifera, which is a rhizophytic macroalgae. And then for each of these control plots, we paired them with a clipped plot where we simulated green turtle grazing. And the paired control and clipped plot were located within 15 centimeters of each other to account for spatial variation. And so we conducted this experiment using seagrass that was already growing, and it just matched these different species identity and richness treatments that we wanted. And so this approach had a couple different benefits. So we were able to conduct the experiment without transplanting seagrass and introducing and so we did not introduce transplant stress, and we wanted to make the conditions as realistic as possible. And so this was a 12-week experiment, and we would travel to the site every 2 weeks to monitor seagrass density or macrophyte density. And we also applied a grazing treatment bi-weekly every 2 weeks for the first 8 weeks of the experiment. And for the grazing treatment, we simulated green turtle grazing by using scissors to clip the seagrass 2 centimeters above the sediment surface. And then for the last 4 weeks of the experiment, we had a recovery period where we were able to look at longer term resilience, and the macrophytes recovered from the grazing disturbance for this period. And then at the end of the experiment, we also collected seagrass biomass cores to look at the biomass in the different plots. And so here are just some photos from the experiment. In this photo, you can see one of our plots. So they were pretty small, and this plot contained 3 different seagrass species. It had Thalassia, Syringodium, and Haliduli. And so when we went out to monitor the seagrass, we would be using snorkels just to count the number of plants of each species in the plots. And then we also would apply the grazing treatment, as you can see here on the right, where we use scissors to clip the seagrass 2 centimeters above the sediment surface. And so at the end of the experiment, which was after the 4-week recovery period, we looked at the impact of the simulated grazing treatment on plant morphology and how it specifically influenced the 4 different seagrass species. And so we found that there was no impact for Halophila. The grazing treatment did not impact height, surface area, leaf width, or number of leaves. And then for Haliduli, we found that the grazing treatment resulted in reduced height, surface area, and width, but no impact on the number of leaves. And then for Syringodium, we found that the grazing treatment resulted in reduced height, surface area, leaf width, and number of leaves. And then for Thalassia, we found the same thing, where the grazing treatment resulted in reduced height, surface area, leaf width, and reduced number of leaves. And so going back to this image, these results match what we expected based on life history traits. So we were looking at the seagrass recovery and the morphological traits at 4 weeks after the disturbance. And so we would expect the pioneer species to have faster recovery from a disturbance, and these species like Halophila were less impacted by the disturbance at the end of the recovery period, whereas Syringodium and Thalassia were more impacted at the end of the recovery period, likely because they have a slower ability to recover from disturbances. And then we also looked at how species richness impacted macrophyte density in the absence of a disturbance. So this data only shows the control plots where we did not apply any disturbances. And we have species richness here on the x-axis and macrophyte density on the y-axis. And we have data from each sampling date, so every 2 weeks. And we did not find any trends between species richness and density on any of the sampling dates when looking at the data without any disturbances. And then when we look at how species richness impacted recovery from disturbances, we see a different pattern. So in these graphs, we have the average richness in the paired plots on the x-axis, and then the paired difference in density as a % on the y-axis. And so a value of -50 would mean that the clipped plot has 50% fewer plants than the control plot. A value of 0 would mean that the clipped and control plot have the same number of plants, and a value of +50 would mean that the clipped plot had 50% more plants than the control plot. And so we found that species richness positively impacted recovery from the disturbance, and we found the strongest trend after the 4-week recovery period, shown here in week 12 on the right. And so we found that a polynomial regression best fit the data, which indicates that there might be a threshold in species richness. And then after that threshold, there might be a positive impact of species richness on recovery from the disturbance, since there was more often more species or more plants in the clipped plot compared to the control plot. And then when we looked at the impact of species richness on recovery after only a 2-week period after the last grazing event, we found that there often wasn't a pattern, with the exception of in week 4, when there is a positive relationship again. So when there is higher species richness, the paired difference in density was closer to 0, indicating that there was a smaller loss in density in the clipped plot compared to the control plot. And these paired plots were more similar when there was higher species richness. And we also looked at the change in the density of individual seagrass species throughout the experiment, focusing on those 3 dominant seagrass species. And so we took the initial density for the species and subtracted it from the final density for the species to get the change in density. And we compared the change in density in the control plots to the change in density in the clipped plots shown on the x-axis. And so for Thalassia, we found that the change in density varied based on the grazing treatment. And so for the control plots, there was they on average had an increase in density throughout the experiment for Thalassia. But for Thalassia in the clipped plots, there was on average a decrease in density throughout the experiment, showing that the grazing treatment had a negative impact on Thalassia density. And for Haliduli and Syringodium, we did not find that the grazing treatment impacted the change in density for these species. And then we also found that the amount of Thalassia influenced recovery after the disturbance. And so in these graphs, we have the percent Thalassia on the x-axis. So this is the amount of Thalassia in the plot. So 100% would mean that the plot only contained Thalassia, and 25% would mean that the plot contained 25% Thalassia and 75% other macrophyte species. And on the y-axis, we have above ground biomass shown in grams. And these outlined points represent control plots, and then the filled in black points represent clipped plots. And so the most interesting finding from this analysis was that there was an interaction between grazing and percent Thalassia. So when the plots were dominated by Thalassia over here on the right, there was a much larger loss in above ground biomass due to the grazing treatment. And when the plots didn't contain as much Thalassia, there was a much smaller loss in above ground biomass due to the grazing treatment. And then this graph shows the same exact trend, but just using standardized data. So now on the x-axis, we have the percent thalassia in the control plot. And on the y-axis, we have the paired biomass. And it was standardized using a log ratio. So we took the logarithm of the ratio of the clipped biomass to the biomass in the paired control plot. And so a value of 0 would mean that the ratio is 1. So the clipped and control plot both had the same biomass. And then as these values get more negative, it means that the difference between the biomass and the clipped plot and control plot is getting greater, with the clipped plot having less biomass than the control plot. And so we found a negative relationship. So when there was a lot of thalassia in the plot, the clipped plot was losing a lot more biomass compared to the control plot. And then when there was less thalassia in the plot, the standardized difference between the paired clipped plot and control plot was much smaller. So this data indicates that areas that are dominated by thalassia might be more severely impacted by grazing disturbances and might lose relatively more seagrass biomass. And so now that we've gone over all of the data, I want to just talk about some of the main trends we found. So overall, we found that the amount of recovery time matters, and we found the strongest positive impact of species richness on recovery after the longer 4-week recovery period. And so the patterns vary based on time because of differences in growth rate and use of below ground nutrient reserves. So longer recovery patterns might be necessary in order to see trends in the relationship between species richness and resilience, and the patterns might change based on the time period used. We also found that the impact of simulated grazing varied based on species identity. So in general, thalassia was the most severely impacted by the grazing disturbance, and we found that thalassia density decreased throughout the experiment in the clipped plots compared to the control plots, and that both thalassia and syringodium had more changes in morphological traits than the other seagrass species. And the plots that were dominated by thalassia also had larger relative losses in biomass. And so going back to the life history traits, this is what we expected. Since thalassia is a longer lived species that has slower growth rates and is expected to have a slower ability to recover from disturbances. And a lot of studies have found that thalassia is able to withstand green turtle grazing, but a lot of these studies were also conducted in more tropical locations. And in the northern Gulf of Mexico, we're reaching the northern range limit for this species. And so these plants are receiving less light, and they're having likely larger swings in temperature, and as well as slower growth rates. So this might make them more vulnerable to grazing or other disturbances, and might be why we saw a more severe impact of simulated grazing on the thalassia. Our results also match what we expected with the insurance hypothesis. So just a reminder, the insurance hypothesis predicts that when different species respond asynchronously to disturbances and fluctuate asynchronously over time and space, this results in increased stability. And we found that the different seagrass species were responding differently to the disturbance, and that when there was higher macrophyte species richness, there was faster recovery from the disturbance, which would lead to increased stability. And so it is likely that less sensitive seagrass species were compensating for losses in more sensitive species like thalassia. And while thalassia was the most severely impacted by the grazing disturbance in this study, a different disturbance or a different environmental context might result in a different species being more severely impacted. And so that's why it's most important to conserve species richness, so that there's the highest probability possible of having a species that is able to withstand given environmental conditions. And so just to summarize, we found that macrophyte species richness may increase ecosystem resilience. So our plots with more species recovered shoot density better than plots with fewer species. And these findings match the patterns that have been observed with seagrass genetic diversity. And we also found that grazing impacts different seagrass species differently. And so areas with more thalassia might have more severe short-term impacts of increased grazing pressure. And therefore, planting and conserving diverse seagrass assemblages could provide a valuable tool for managing seagrass beds and maintaining seagrass stability. And so these concepts can be incorporated into management and restoration in a few ways. So they can provide insight into the vulnerability and resilience of different seagrass meadows. And specifically, this study was conducted in Crystal River, Florida, which has high boating traffic. And we've discussed in previous webinars how there are also a lot of propeller scars in the region. And so in the study area, species richness might actually be a really important tool because it might be able to speed recovery from disturbances like propeller scars. And adding species richness to seagrass monitoring programs could also provide insight into changes in disturbance regime, successional stage, or the stability of the ecosystem, with the implications varying based on environmental and biotic context. And so, like I said, this research was conducted as part of my dissertation at the University of Florida, but I'm really interested in following up on these research findings in the future. And so when I start my postdoc position at Mote Marine Laboratory, I will be researching ways of increasing seagrass restoration success. And so I'm interested in exploring how to incorporate resilience into seagrass nurseries and outplanting for restoration, and specifically exploring how species richness and genetic diversity impacts Florida's seagrass restoration. And so this project was a team effort and would not have been possible without the help from all of these people and without funding from the Nature Coast Biological Station and the Florida Sea Grant. And so thank you to all who have helped with this, and thank you all for joining. And I can take any questions, and feel free to reach out to me via email.
Thank you, Jamila. That was a great presentation. If anyone has questions, please, you're welcome to enter them into the chat, or you're also welcome to unmute yourself, turn your video on if you like, and ask those questions. I have a question while people are sort of thinking about things. The first is you presented some really convincing data that mixed meadows increase resilience. We think about seagrasses not just about how resilient they are, but how many species they support. Is there any evidence? Are there any downsides to multiple species meadows? Do those meadows that have multiple species, do they have the same number or more or less organisms living in them?
That's a really good question. I know that, like I said, like having higher genetic diversity can increase the invertebrates, since there's more structural complexity, which provides protection from predators. So I would expect to have the same trend with species diversity, where there's more niches and more protection from predators. And it is something that I looked into a little in my dissertation, and did find that in some samples that contained 2 seagrass species, there was higher invertebrate diversity and density. So I would think in general, they probably support just as many or more animals.
Cool. Brad, did you have a question?
Yeah. Well, great talk. I really enjoyed it. I was thinking about the callirpa you mentioned earlier in your slides. Did you see any response of callirpa to the simulated grazing experiment?
So we were not measuring callirpa height, but I think that we saw that it was pretty resilient to the grazing. It's like one of the most early successional, or it occurs early successionally. And so I would expect that it recovers really quickly from disturbances, and it's something that would be interesting to look at specifically how the macroalgae was responding.
We have a couple of questions popping up in the chat. The first is from Clyde Bunting and says, moving south to north along the Gulf Coast, does plant diversity change?
I am not sure if there are any general patterns, but I know there are like 4 or 5 seagrass species in Crystal River and in Cedar Key, and I think those continue further south. But
yeah, that's true.
Yeah. Okay.
The next question is from Trisha, and it says, how much regrowth, if any, were you seeing in Thalassia blades in the 2-week period after being clipped?
So we were seeing pretty fast regrowth in all of the species. So they were, don't remember the exact numbers, but it was almost hard to tell which plots had been clipped. They were going back to like 10 centimeters or more, I think, over the 2 weeks. So there was pretty high regrowth in all species.
And we have another question from Mike Allen, which says, great job, Jamila. I had a question about the role of intra versus inter-specific competition and how that could have influenced your results. For example, I wonder if the treatment responses showing benefits of multiple species could be a release from inter-specific competition in sites with only one species.
Yeah, that's really interesting. And it's something I've thought about a little. And I think, so we didn't see any impact of species richness in the absence of disturbances. And so we would expect if it was a release from intraspecific competition that there would be increases in density perhaps without us applying disturbances, but there is a lot of noise and variability in these ecosystems. So it's possible we just didn't see that trend, but the impact or the positive impact of species richness when there was when we applied the disturbance could show it fits with the stress gradient hypothesis, or where there's more positive interactions when there's higher stress, and so they might be kind of released from those competitive interactions when there's a disturbance or stressful condition.
Anybody else have any questions? Mike says, thank you.
Yeah, thank you.
I have one more question. You talked I mean, in this instance, you applied the stressor, the grazing, directly to all of your species. Would that happen in nature? Would these grazers go for monospecific meadows? Would they go towards multi-species meadows? How does multiple species affect how much grazing happens?
Yeah, that's definitely a way that we simplified natural conditions in the experiment. And I think it's a little bit unknown, but since these are not known to be like highly specialized grazers, I would expect that they would seek out multi-species meadows or wouldn't be deterred by multi-species meadows. And I did look into that a little bit for sea urchins and found that they weren't deterred by multiple species assemblages. And so by feeding from a couple of different species, we would expect the herbivores are trying to maximize their nutritional intake and minimize toxin intake. And so I would expect that they wouldn't be deterred by multiple species meadows. And looking into data just from like green turtles, different studies find that they prefer different seagrass species. So in some studies, they look like they primarily eat Thalassia, where other studies show manatee grass, Syringodium, or haliduli, or even macroalgae as being their main diet. So it seems like they eat a variety of different macrophytes.
Cool. Anyone else?
hot topic right now. There's been a lot of urchin grazing around Steenhatchee and Keaton Beach area. So I just wonder, this study was about simulated turtle grazing, but I wondered, since you have worked with urchins, what your thought is on, if you had simulated urchin grazing, what the results would have looked like.
Yeah, that's interesting. I think it would have been similar. I think when there's like when there's overgrazing by urchins, when there's really huge populations, they consume most of the above ground biomass and would have similar impacts. But it would be something to look at how the seagrass responds to different types of grazers. And I have compared a little bit how different types of grazing impact different seagrass traits and anti-herbivore defenses, and found that there were pretty similar impacts of the urchin grazing to the simulated green turtle grazing, although the simulated green turtle grazing did impact some chemical traits that weren't impacted by the urchin grazing. So following simulated green turtle grazing, there were increases in leaf phosphorus, for example, where I didn't see that pattern with the urchin grazing. So it might just impact future grazing events, since the green turtles might alter leaf chemistry in a way that makes the leaves more desirable for herbivory in the future. But it's definitely something I'd be interested in exploring.
Thank you.
Yeah, thank you.
We have another question in the chat. Says, really fascinating, Jamila, and I'm wondering whether there's a phylogenetic signal of body shape, size, masking biomass, productivity response. For instance, did you consider correcting for differences in body shape between seagrass species? The reason I ask is it seems to me that clipping 2 centimeters of thalassia removes considerably more body mass than either Syringodium or Haliduli.
Yeah, that's an interesting point. It is not something that I really incorporated into the experimental design, since we were kind of doing a standard grazing treatment, since green turtle grazing can just remove all above ground biomass from the macrophytes. But it is something that definitely influences recovery. And so we were clipping to 2 centimeters above the sediment surface, not 2 centimeters. And so we were removing the above ground biomass basically for all of the species. And but this body morphology, and specifically below ground morphology, can influence recovery, since these larger species like thalassia have larger stores of nutrients and carbohydrates below ground, which they can mobilize and use these stored nutrients to regrow following grazing. And so this differences in body mass definitely influence patterns in recovery.
Cool, thank you. Anyone else? Well, I think questions have slowed. So I'd like to thank you again, Jamila, and thank everyone else for the really nice discussion afterwards. Those were some really good questions. But I think we'll let you go, and we hope to see you back next month to hear about carbon in seagrass meadows. So thanks again, y'all.